Alzheimer's is one of the most prevalent neurocognitive diseases in the world. Globally, over 57 million people have Alzheimer's, and over 120,000 deaths are related to the disease. This year alone, over $380 million are predicted to be spent on Alzheimer's care in the United States. Nutrition is a contributor to the progression and onset of Alzheimer's disease. One contributor to the disease can be the effect of excess sugar on the brain. The brain uses 20% of your energy. However, it is very picky in terms of energy source, only wanting glucose. When you have too much intake of sugar, the brain ends up not using it all for energy and becomes excess sugar in the brain. These free glucose molecules, not being used for energy, lead to protein glycation. When this happens, the protein structures are changed, which can impair the function of the protein. These are called advanced glycation end products, or AGEs for short. AGEs can lead to chronic inflammation, cytokine release, reactive oxygen species production, and blood vessel and neuron damage. AGEs further lead to many of the findings characteristic of an Alzheimer's brain, such as amyloid plaques, tau tangles, synapse loss, neuron death, inflammation, vascular damage, and brain energy failure. Formation of AGEs can be directly combated by diet improvements. First, we should begin by decreasing sugary food intake, especially high fructose corn syrup products. After that, we should consume foods that do not generate AGEs. In your diet, you should increase the amount of fresh fruit, vegetables, whole grains, legumes, nuts, seeds, eggs, lean meats, and fish. More foods to increase in your diet are berries, the darker the better, dark grapes, turmeric, cinnamon, dark leafy greens, garlic, onions, and dark chocolate. These foods reduce glycation and neutralize AGEs, which can possibly improve brain function and prevent the onset of Alzheimer's. Studies have shown that alcohol is a contributor to the development of Alzheimer's and dementia. Brain scans have shown that the brain shrinks with alcohol, showing similar atrophy to those with dementia. Alcohol also leads to decreased brain activity. Another impact of alcohol on the brain is through the body's production of acetaldehyde during alcohol metabolization. Acetaldehyde is a toxin, and accumulation can lead to increased inflammation and death of neurons. Alcohol becomes acetic acid in the body, which can produce fat, and studies have shown obesity to be a contributor to Alzheimer's. Alcohol also reduces thiamine absorption, which is also found in Alzheimer's patients. The effects of alcohol are so great that there has even been a new condition termed alcohol-related dementia, a form of dementia caused by excessive alcohol consumption. Another substance associated with Alzheimer's development is Adderall. Adderall causes dopamine surge in the brain, serotonin increase, and abnormal activation of brain circuits, and causes the loss of brain cells. Long-term abuse of amphetamines, like Adderall, may accelerate neurodegenerative processes. Furthermore, illicit Adderall use is associated with increased use of other drugs like alcohol, which we know can cause a lot of neurological damage. Importantly, this research mostly pertains to individuals taking Adderall illicitly, and not due to diagnoses such as ADHD. In fact, for individuals with ADHD, Adderall and other medications might mitigate the risks of developing dementia. Lastly, illicit Adderall use is also associated with poor diet quality, possibly causing AG formation. It's important to stay away from illicit drugs and alcohol to ward off the development of Alzheimer's. Another contributor to Alzheimer's disease is nutrition and its effects on gene expression. Nutrigenomics is the study of how our diet can affect our gene expression. The nutrients that we get from our diet can affect our gene expression in 3 different ways: through nuclear receptors, metabolism, and signal transduction. The most common finding in individuals with Alzheimer's disease are extracellular neuritic plaques. These plaques are made of amyloid beta peptides, which are commonly found in the brain. The reason for their aggregation in the brain can be one of 3 things: overproduction, insufficient clearance, or defective proteolytic degradation. The release of amyloid beta peptides in the brain is due to a process called the amyloidogenic amyloid precursor protein, or APP processing pathway. This pathway is initiated by a series of enzymes and proteins whose expressions have the possibility of being turned on or off, possibly due to one of the processes we mentioned earlier. A diet with reduced vitamin B12 content has been linked to overproduction and dysregulation of this important pathway. Vitamin B12, along with folate, is essential in converting homocysteine to methionine, which ensures that the levels of homocysteine in the body won't get to dangerously high levels. When homocysteine levels are too high in the body, it can lead to a variety of health issues, including cardiovascular disease, blood clots, and general oxidative stress in the body. High levels of homocysteine in the cerebrospinal fluid have been linked to patients with Alzheimer's disease, showing that the consumption of vitamin B12 has a decreasing effect on the development of Alzheimer's disease. You can increase your vitamin B12 intake by consuming an adequate amount of red meat, fish, and dairy products. We recommend adhering to the Mediterranean diet to ensure consuming healthy foods rich in antioxidants and staying away from drugs and alcohol that will decrease the risk of developing Alzheimer's.